Imagine a European robin flying through just a pitch black sky.
Yeah, no stars, no landmarks at all.
Right. It's thousands of miles from its breeding grounds. It can't see the ground, and yet it navigates with this like absolute pinpoint precision.
It's incredible. And for the longest time, we thought biology was just this warm, wet, chaotic soup.
Like just chemical reactions bumping into each other randomly until something finally clicks.
Exactly. We viewed it as a machine governed entirely by classical thermodynamics, just Newtonian physics on a tiny scale.
But this new research we're looking at today proves that this bird, it isn't just relying on classical physics. Deep inside its eye, there are these subatomic particles doing a literal quantum dance that lets the bird, well, basically see the Earth's magnetic field.
Yeah, it completely dismantles the textbook version of biology we've relied on for decades. Underneath all that classical chaos, your cells are actually operating as these astonishingly efficient quantum machines.
Which brings us to our mission for this deep dive. We've got a massive stack of research today that is going to completely rewire your understanding of life itself. We're exploring the proven mechanisms showing how biology relies on quantum mechanics. But, okay, wait. Haven't physicists always said that quantum states are incredibly fragile?
Oh, absolutely.
Like, don't quantum computers need to be locked inside absolute zero freezers just to work?
They do, and that was the prevailing dogma for a really long time. I mean, if we rewind to 1944, Erwin Schrödinger.
Oh, the guy with the cat-in-the-box thought experiment.
That's the one. He actually published a book called What is Life, where he hypothesized that life's fundamental mechanisms had to be rooted in quantum order. But, the biological community pushed back hard on that.
Because life is too hot and messy.
Right. They pointed to decoherence. The idea is that the chaotic thermal energy of a biological system would just instantly destroy any fragile quantum state.
So they basically laughed it off. But then, and this is where the modern research comes in, we developed tools like ultrafast spectroscopy, right?
Yeah, which lets us watch molecules move at the femtosecond scale. That's 1/4,000,000th of a second.
It's mind-blowing. And we realized Schrödinger was actually right. So to help you listening navigate this immense stack of research, I found myself organizing these quantum phenomena into a mental framework. Just 3 letters: C-T-E.
Coherence, tunneling, and entanglement. I like that.
Yeah. Let's unpack the C first. Coherence. From what I understand, this is basically the story of how life gathers and absorbs energy, right?
That is the perfect starting point. So quantum coherence is about phase correlation between quantum states. Let's look at photosynthesis.
Okay.
Specifically in green sulfur bacteria. They have this structure called the Fenner-Matthews-Olsen complex, or FMO for short. When a photon of sunlight hits this complex, it creates a little packet of energy called an exciton.
And that exciton has to travel through a dense network of pigment molecules to reach a reaction center, right? Like to turn into actual food.
Exactly. Now, classically, you'd imagine that exciton acting like a delivery driver stuck in city traffic.
Driving down one street, hitting a dead end, turning around, just a random walk.
Right, which would be incredibly inefficient. And the classical model predicted exactly that kind of pinball hopping. But when researchers used two-dimensional electronic spectroscopy, firing these super short, synchronized laser pulses at the complex, they didn't see a random walk.
They saw a quantum wave.
Yes. The exciton enters a state of quantum superposition. It isn't just taking one path, it's literally exploring every single possible pathway through that molecular network at the exact same time.
So it's like a wave of water instantly washing through a maze, instantly finding the fastest route to the exit, rather than a single rat trying to run through it.
That's a great visual. Because it acts as a coherent wave, it finds the path of least resistance instantly. The quantum efficiency is greater than 90%. Almost zero energy is lost.
Okay, let's unpack this because I have to push back here. We just talked about decoherence. How does this super fragile quantum wave survive the boiling heat of a living cell?
Right. Shouldn't the noise instantly collapse it?
Yeah. How does it survive?
This is honestly the most brilliant part of the mechanism. It's called environment-assisted quantum transport, or ENIQT. You're right that random noise destroys quantum states, but the noise inside the FMO complex, it isn't random.
Wait, what do you mean?
The protein scaffold holding those pigment molecules together vibrates at highly specific, tuned frequencies.
So the protein isn't a chaotic blender. It's more like a tuned musical instrument.
Exactly. It's called vibronic coupling. The protein's vibrational energy actually resonates with the exciton. Imagine a surfer on a wave. If the water is totally still, the surfer eventually loses momentum and sinks.
Right.
But the thermal vibrations of the protein provide constant, gentle little kicks that keep the excitant moving. The heat and the noise actually stabilize the coherence.
Oh, wow. So life didn't just survive the noisy environment, it literally evolved to use the noise.
To its advantage, yeah.
Okay, before we jump to the next letter, let's lock this in. Think about your own daily energy slumps. What if your cells could perfectly map out energy distribution without losing a single drop, using the very heat of your body to serve multiple pathways at once? It's beautiful.
It really is.
But gathering energy is only half the battle. How does life actually use that energy to drive biological changes? Which brings us to the T in our CTE framework, quantum tunneling.
Right, so if coherence is the wave, tunneling is the ghost. Quantum tunneling happens when a subatomic particle passes directly through a solid energy barrier, a barrier that, classically speaking, it absolutely doesn't have the energy to cross.
Okay, so to visualize this classically, if a chemical reaction is a ball trying to roll up a huge hill and it doesn't have enough speed, it just rolls back down. Reaction denied.
Exactly.
But with quantum tunneling, the ball just phases directly through the solid center of the hill and pops out on the other side, like walking through a brick wall.
A perfect analogy. And this ghosting happens millions of times a second in your enzymes. Take oxidoreductases, like alcohol dehydrogenase.
The enzyme that breaks down alcohol in your blood.
Right. Its job is to strip hydrogen atoms off the alcohol molecule. But when scientists measured the speed of this reaction, it was happening vastly faster than classical thermodynamics would permit.
Because the hydrogen wasn't climbing the hill, it was tunneling through it.
Yeah.
But, and this is a big question, how do we actually prove that? How do you prove a single atom is ghosting through a wall?
We use a beautifully elegant mechanism called the kinetic isotope effect, or KI. Hydrogen is the lightest element, 1 proton, 1 electron. Researchers swapped that hydrogen atom with an isotope called deuterium.
Which is basically heavy hydrogen, right? It has an extra neutron.
Exactly, making it twice as heavy. So classically, if you're just rolling a ball over a hill, making it twice as heavy might slow it down a bit, but it should still make it over. But in the quantum realm, every particle has a wavelength. It's de Broglie wavelength. The heavier a particle is, the shorter and tighter its wave becomes. Because deuterium is heavier, its quantum wave function is compressed. It literally loses its ability to reach through the energy barrier.
Oh, wow.
So when they tested the heavy isotope in the enzyme, the reaction rate just plummeted far beyond what classical physics predicted. The heavy isotope simply couldn't tunnel.
That is staggering. So enzymes exploit quantum mechanics just to keep us alive. But based on our research stack, tunneling isn't just about breaking down chemicals, it's fundamentally altering our genetics, isn't it? Let's talk about DNA shape-shifting.
Yeah, this is where it gets really deep. DNA is held together by hydrogen bonds. You know the classical base pairs guanine pairs with cytosine, adenine pairs with thymine?
Right, the letters of our genetic code.
Exactly. The physical bridges holding those letters together are made of protons. But because of quantum tunneling, those protons don't always stay put. They can spontaneously tunnel across the barrier to the other side.
Wait, meaning the actual physical shape of the DNA letter changes?
Yes, it shifts into a transient tautomeric form. So the normal keto shape of a base shifts into an enol shape. Amino shifts into imino. Usually, this only lasts for a fraction of a picosecond before the proton goes back.
Okay, but timing is everything, right?
Yeah.
What happens if the cell is dividing and the DNA replication machinery unwinds that exact section of code during that split microsecond when the proton is on the wrong side?
Then the biological machinery reads the wrong shape. It looks at a guanine that's temporarily masquerading as an adenine, and it mispairs it.
And a mutation is born.
A spontaneous point mutation, exactly. And we have immense clinical evidence for this. Look at the KRAS and TP53 genes.
Which are massive oncogenes. I mean, TP53 is basically the guardian of the genome. It's heavily implicated in human cancers.
Right. And researchers found that these specific genes have highly susceptible oncogenic hotspots where this proton tunneling frequently occurs. The proton ghosts across, the DNA mispairs, the protein misfolds, and suddenly you have runaway cancerous division.
That presents a wild philosophical paradox. If quantum tunneling causes random mispairings that lead to things like cancer, then quantum mechanics is a driver of disease. But mutations are also the engine of evolution.
It is the ultimate biological double-edged sword. If DNA were just a rigid classical ladder, we wouldn't have the mutation rate needed for adaptation. The same subatomic ghosting that gave rise to human complexity also introduces fatal errors.
Okay, let's pause for a quick 5-minute recap to anchor this.
Yeah.
Knowing your DNA is this vibrating, shape-shifting quantum structure really changes how you see genetics,
For sure.
So we've looked at classical versus quantum biology. We covered coherence excitant, surfing multiple paths in photosynthesis. And we just covered tunneling protons, ghosting through walls, driving enzymes, and causing DNA mutations. But that's all inside the cell. How does life sense the outside world? That brings us to the E, entanglement.
Entanglement. Probably the most famous quantum phenomenon. It's when two particles become so deeply linked that the state of one instantly correlates with the other, no matter how far apart they are. In biology, this sensory interface operates through the radical pair mechanism.
Here's where it gets really interesting. Let's unpack this carefully. People usually explain entanglement with that cliche of two magic coins spinning in different cities that always land on the same side.
Right, which isn't super helpful for biology.
Exactly. So let's get into the physics. How does this let the European robin from our intro navigate?
It starts in the bird's retina. They have a highly specialized protein called cryptochrome. When ambient blue light hits this protein, a photon knocks an electron completely out of its molecular orbit.
Okay.
This creates a radical pair, two distinct molecules that each have an unpaired electron. Because they were created together by the same photon, their quantum states are entangled.
And what exact property is entangled?
Their spin. Spin is a fundamental quantum property. These two electrons can exist in one of two states. Parallel spins, called a triplet state, or anti-parallel spins, which is a singlet state.
And this is where the Earth's magnetic field comes in, even though it's incredibly weak. Like, down to 50 microteslas, that's way weaker than a fridge magnet.
It's absurdly weak, but it's enough to tilt the quantum axis. As the bird turns its head, the angle of the Earth's magnetic field interacts with the spin states of these entangled electrons. It constantly toggles them back and forth between singlet and triplet.
Okay.
And here's the biological translation. The singlet state and the triplet state produce entirely different chemical reactions in the retina.
Wait, are you saying a bird literally sees the magnetic field overlaid on its vision because of entangled electrons in his eyes? The magnetic field changes the spin changes the chemicals, and the optic nerve reads that.
It's a literal real-time quantum sensor, and it doesn't stop at visual. There's impelling research into the vibrational theory of olfaction, too. Yeah. The theory suggests your olfactory receptors aren't just identifying the physical shape of an odor molecule like a lock and key. They might actually be using inelastic electron tunneling to measure the specific quantum vibrational frequencies of the molecule. You are literally smelling quantum vibrations.
That is insane. If birds can sense magnetic fields through quantum spin and we smell via tunneling, what hidden senses might humans have lost or maybe still possess without realizing? It's wild to think about.
It really is.
But we need to shift gears. If our biology is built on this impossibly delicate quantum balance, what happens when it breaks down? What happens when the noise finally wins?
That leads us to the pathology of quantum biology, mitochondrial decoherence.
Okay, the power plant of the cell.
Right. Inside the mitochondria is the electron transport chain. Specifically, let's look at the FMN to iron-sulfur axis. To create ATP, the energy keeping you alive, electrons have to travel across a series of redox centers. But the physical gaps between these centers are too wide for classical physics. The electrons must use quantum tunneling to cross, and they rely on partial quantum coherence to synchronize their flow.
I visualize this like a precisely timed biological bucket brigade. The redox centers are workers, passing buckets of electrons down the line to a perfect beat.
That's a great way to look at it. But remember how we said the FMO complex uses structured noise to maintain coherence? If a cell experiences extreme stress, toxins, or just the structural disorder of aging, the noise stops being structured. It becomes chaotic.
And the system experiences decoherence.
Exactly. The quantum phase relationship shatters.
So someone in the bucket brigade loses the rhythm and a bucket gets dropped.
And a dropped bucket here is a leaked electron. When an electron leaks out, it immediately reacts with nearby oxygen, creating reactive oxygen species, or ROS.
Free radicals.
Right. A tiny amount of ROS is normal. But massive quantum decoherence causes a flood of free radicals. They tear through the cell, ripping electrons off DNA and proteins, causing catastrophic oxidative damage. This loss of quantum order is the root mechanism driving aging, metabolic dysfunction, and cancer.
Wait, so aging isn't just biological wear and tear, it's literally a measurable breakdown of quantum physics in our cells.
It's entirely measurable. Clinical researchers have used ultrafast spectroscopy to compare the mitochondria of aggressive cancer cells to healthy ones. They found that the quantum coherence lifetimes in the cancerous mitochondria were measurably shorter. The quantum timing belt had snapped.
Wow. Okay, time for our second 5-minute recap. We just covered entanglement, how radical pairs act as magnetic compasses in cryptochromes. And we covered decoherence, how failing quantum systems in mitochondria cause aging and cancer. If aging is just a loss of quantum rhythm, could we one day tune ourselves back to a younger frequency? Which begs the question, how do we actually see this stuff to fix it?
The technological leap recently has been staggering. We've moved from theory to direct observation. We already mentioned 2DES, two-dimensional electronic spectroscopy, catching quantum beats with femtosecond lasers. But we also have entirely new diagnostic tools like NV centers and diamonds.
NV centers. Nitrogen vacancy defects. I've seen this in the literature. How does a diamond act as a biological sensor?
Imagine a microscopic diamond lattice, just carbon atoms. If you swap out one carbon atom for a nitrogen atom and leave a microscopic hole or vacancy next to it, that defect traps an electron.
Okay.
The quantum spin state of that trapped electron is absurdly sensitive to its environment. We can inject these biocompatible nanodiamonds into a living cell. The cell ignores them, but we can read the spin of that trapped electron to map the exact magnetic fields and localized ROS free radical flux inside the cell with nanoscale precision.
So what does this all mean? We're basically injecting nanoscale radar dishes into the cell to track the quantum weather, but what about the computational side? If we want to design a drug to fix this, our computers need to be able to do the math. And we are currently in the NISQ era, noisy intermediate scale quantum computing. Are our computers actually powerful enough yet to invent quantum drugs?
It's challenging, but we're crossing the threshold. Classical supercomputers simply cannot calculate the deeply entangled electron states of a complex protein. But by combining cryo-electron microscopy with quantum algorithms, specifically the variational quantum eigensolver, or VQE, we can run quantum machine learning protocols.
Which allows us to do what?
VQE algorithms let us map the precise electron delocalization of a protein. We can simulate exactly how a TP53 mutation alters the quantum folding landscape of the molecule, something a classical computer just can't handle.
Which opens the door to quantum-informed medicine.
Exactly. We are moving past just mopping up chemical symptoms. Imagine diagnostics where measuring the coherence lifetimes of your mitochondria is a primary biomarker for biological age.
And for therapeutics. If proton tunneling is causing DNA mutations, could we theoretically design a drug like a DNA stabilizing agent that subtly alters the water molecules around the DNA just enough to widen the energy barrier and stop the tunneling?
You're touching on the bleeding edge of pharmacology. Modulating the hydration shell to alter tunneling probabilities, using tailored magnetic fields to target radical pairs, or designing redox modulators specifically to restore coherent electron flow in the mitochondria. We are literally learning to tune the quantum frequency of the body.
Think about that for a second. Would you take a medication that doesn't just change your chemistry, but fundamentally alters your subatomic physics? It is a wild reality to consider. As we bring this deep dive to a close, let's do a final recap. We've looked at the tech revealing this world 2DES, NV centers, quantum computing, and the future of diagnosing and treating disease at the quantum level. But let's look at the ultimate horizon of the science.
Yeah, if we connect this to the bigger picture, there is one hypothesis that attempts to explain the greatest biological mystery of all, the Penrose-Hameroff orchestrated objective reduction hypothesis, or ORCH, or R.
This is the one proposed by Nobel laureate physicist Roger Penrose and anesthesiologist Stuart Hameroff, right?
Yeah.
Unpack this for us, because if this is true, it changes everything.
It does. They suggest that the protective mechanisms we've discussed, like inacute shielding coherence from noise, are operating at an incredibly high level inside the human brain. They hypothesize that inside our neurons, there are structural filaments called microtubules.
Okay.
These microtubules are supposedly capable of isolating and sustaining massive quantum coherent states. And crucially, they propose that the physical collapse of these quantum superpositions into a single state is the actual fundamental mechanism that generates human consciousness.
That is staggering. So if our neuronal microtubules are actively processing quantum superpositions, quantum mechanics isn't just the engine of our metabolism. It might be the literal origin of free will. Our thoughts, our memories, they could all be biological quantum computations happening in the warm, wet brain.
It remains one of the most fiercely debated hypotheses in science. It needs way more empirical verification, but the very fact that it's scientifically plausible highlights our ultimate trajectory. Life is not an accident occurring in spite of quantum mechanics. Life is an intricate, evolved expression of it.
So let's bring it all together. Next time you feel the sun on your skin or take a breath or simply think a thought, remember what is happening just beneath the surface. You aren't just a biological machine made of meat grinding away on classical gears. You are a walking, talking quantum computer. A marvel of coherence, tunneling, and entanglement, which leaves you with one final question to ponder. If your consciousness is truly quantum, does that change how you define life itself? Keep questioning the visible world around you because the real magic is happening at the quantum scale.